opportunity to talk and for all of the organization that happened ahead of this talk. So as Miko said, I am a research fellow at CREAM at the University of St Andrews, but the work I'm presenting today is part of several projects that I've done over the years when I was part of the University of Cape Town and also Nelson Mandela University, and it's been funded by the Royal Society, the National Research Foundation in South Africa, and yeah, I think those two. Okay, great. So what I'm going to talk about today are hidden Markov models for movement ecology, and I had a look at what's coming up in your seminar series in the next few months, and it sounds like you're going to be hearing more about movement ecology, and in more general terms, so not just HMMs. So HMMs have become very popular recently for analyzing movement data, but they're by far not the only way one can analyze animal movement data. Okay. So goals for today, I'd just like to give a brief introduction to, and it will be very brief, to hidden Markov models, and hopefully give you an idea of how they work, explain a little bit about their limitations, and convince you that they're actually quite versatile and fun models. So the kinds of HMMs I'm going to be talking about today are frequentist ones, where we use numerical maximization of the likelihood to get at the parameter to estimate the parameters of interest. One can implement HMMs in R using a range of packages. So these three that I've listed here, Move HMM, Momentum, and HMM TMB are very good places to start. The first package, I'm not sure if you can see my cursor, but Move HMM only takes in location data, so X and Y locations, but Momentum and HMM TMB are very flexible and can be used with all sorts of kinds of data. You can also write HMMs from scratch in R, so that you can also write them with bespoke code. You don't need to use a package. So generally, when one analyzes animal movement data with HMMs, you assume that your data has little or no error. You can model it, but that the sort of vanilla flavored HMMs assume no error in locations. We also need observations to be made at regular intervals, time intervals. This is a requirement of the model, the model mechanisms. And in this talk, I'm not going to talk about random effects. Sorry about that if you were hoping for random effects, but you can fit models with random effects in Momentum and HMM TMB. Okay, so hidden Markov models are flexible general purpose models for time series. They're not something that's used exclusively for animal movement modeling. They are used in all sorts of kinds of areas of research. So what's different about a hidden Markov model compared to, say, a regression model is that assuming that there's this underlying unobserved process that generates observations. So we have observations, and what we want to get at is this unobserved underlying process. So we call this the state process. And the state that this state process can be in is described using a statistical distribution. So we take an observation, we say, which distribution of finite number of distributions is this observation likely to have come from? And the underlying process is assumed to be a Markov chain. A Markov chain, if you haven't come across this term before, it's called a chain because it's a sequence of possible events. And a sequence of random variables is a discrete time Markov chain if it satisfies two conditions. Number one, there's a fixed number of states, and the Markov property holds. The Markov property is a way of describing how new values evolve, so how new values arise, how new values depend on the history of the process. So basically, we can describe this as conditional on the current state, the future is independent of the past. So we only have this one time step dependence going forward. We no longer assume independence between subsequent events, but we'd only assume a one-step dependence in the most simple HMM, and the standard HMM rather. So this is a classic picture of an HMM where we have in the top row, in the bold Zs, we have the state dependent process, and these are the observed variables. And they're bold because it could be, it's normally a vector. So it's often X and Y locations, or it could be X and Y and Z if you've got an animal that dives or flies. And it could be a location plus other information that you have from a tag, for example. And in the lower process, where I've denoted them with C's, that is the state process, and that's hidden. So that's the process we typically want to know about, but don't know. So all we have are the observations, and we use this model structure to make inferences about the underlying states. So that's the part that I said. There are a fixed number of states. When we fit an HMM, we choose the number of states. That's what I mean by finite possible values. So when we fit an HMM, we say, okay, I think I'm going to try fitting a model with 3 states. So in this case, n can be 1, 2, or 3. And we have serial dependence in the state process. So that's why there are arrows connecting the C's. But there are notice there are no arrows connecting the Z's. And that's because the value of C selects which of the n component distributions generates an observation Z. The observations Z are independent, conditional on the underlying state. So we don't have dependence between subsequent observations, but they are independent, only conditional on the underlying state, which does have a dependent structure. So let's think of a quick example. This is a non-movement example. Say that we have some observations. Let's put some values to the observations. Here, these are temperatures. And let's say that we have we have 2 states in the state process, 1 or 2. So the state that the process is in will select which of the component distributions, which of the 2 component distributions will generate an observation. Here, the observations are temperature. So say our underlying states are the weather is in a warm spell or a cold spell. So warm spells will generate warmer temperatures than cold spells will. But we don't normally know what state it's in. We can make we can use the model structure to make inferences. So we can ask questions like, what weather regime does each observation come from? What do the generating distributions look like? And how often does the process switch state and when? So this is a slightly different way of thinking about it, and this includes like all of the parts of an HMM. So let's start on the left-hand side. So we have a Markov chain, and we have 2 states, and the delta 1 and delta 2 tells you how much time or how much time the process spends in each state. So here we have a process that spends most of its time in state 1, so 0.75. So these 2 things need to add up to 1. So in this case, we have a process that's spending most of its time in state 1, 75% of its time, and 25% of its time in state 2. So moving just down the column where it says Markov chain, you can see that there are black dots and open dots. Black dots denote where the process is. So here the process is starting in state 2, and it's got a probability of moving to state 1 or staying in state 2. So we've got, these are called the transition probability. So we've got a transition probability of staying in state 2 to state 2 of 0.7, and moving from state 2 to state 1 of 0.3. So, and then in the next time step, you see the process moves into state 1, and then from there we've got a probability of staying in state 1 of 0.09, or probability of moving back into state 2. So those are the transition probabilities. So if we move to the right, then we can see we've got these 2 state dependent distributions. You can see the curves in the top where we've got P1x and P2x. So these are the underlying distributions that we generally want to make inferences about. And you can think of these as basically generating an observation. So an observation belongs to one of these 2 distributions. So if we start with the first observation, 31.1, and you can see there's a little tick mark just above 30, and that you can see that falls quite squarely into that second distribution. It's very unlikely to have come from the first distribution because there's basically no mass there under the first distribution. It's gone to 0. And then moving back down, we've got an observation of 16.3, and that looks much more likely to have come from the first distribution, and it carries on that way. So this kind of it gives you an idea of what we're assuming is going on in the background of an HMM. So as with any method, there are opportunities and there are limitations. So so here are some things that you can and can't do with HMMs. So with HMMs, you can estimate some really useful quantities that help you learn about your animal movement. For example, the most likely number of states, and this is how you do this is outlined in a paper by Jennifer Poehler et al. In 2017. And essentially, you can't just blindly follow the AIC. You need to exercise a little bit of common sense and have knowledge about your study system. Typically, the AIC will ask you for more and more states. So that's why you can't just blindly follow the AIC and keep adding states. You can also find out the shape of the distributions that the observations come from, if they're very peaked, if they're very flat, and you can also find the most likely state that corresponds to each observation. And there are two processes to this. You can either use global decoding or local decoding. Global decoding takes into account the whole sequence. So what is the most likely sequence of states in this whole time series, whereas local decoding is much more about, okay, which state is this observation most likely to come from? And you can also figure out the probability of switching between states, and these are the transition probabilities that I was talking about in the previous slide. So what you can't do with HMMs is find out what the movement states mean, and this is really important, and it's a hard no, you really can't. HMMs are totally blind to what the data are, and they're basically just fitting to the data themselves without any sense of what those data actually, where those data come from, what metrics they're actually, where they actually are. So you can't find out what the movement states mean. You also can't fit models with loads of covariates. And this is a sort of not really, because there's no automated way of doing model selection with HMMs yet. So it basically is very labor intensive, and you might lose sight of what you're actually doing if you've got lots and lots of covariates. A lot of people say, well, my process might have lots and lots of states instead of one or two or three, or instead of two or three. Can I fit a model with lots and lots of states? And the answer is yes. Yeah, and I've put here yes and no, because it's no because you can't do it with the standard machinery that you'll find in the packages that I listed, but you can do it if you code the models up yourself and add some clever maths to deal with the fact that you'll end up with really large matrices. And the fourth thing you can't do is fit models to irregular data. And this is a sort of yes, well, yes, actually you can. And this is if you model the irregular data first and then regularize the tracks and then fit the HMM. So a paper came out in 2022 that I was involved with by Richard Glennie, and the paper was called Hidden Markov Models, Pitfalls and Opportunities in Ecology. And here we list some of the interesting things that one can take forward with HMMs and develop further. So the sort of research avenues that people might be looking at going forward. But it's an interesting read if you already know about HMMs and you're like, well, what other things can they do? Or if you've already been thinking about some of the limitations that they have. Okay, so things to remember here. States are only interpretable if we have meaningful state-dependent variables. If you've got something that you don't know how to interpret, it's going to be very difficult to make sense of any model results. The states identified by the model might not make biological sense, and that's something to keep in mind. Yeah, you might not be able to, you might have some movement state that doesn't actually correspond to anything functional that the animal is doing. And another thing to remember is that often we're using spatial data to fit HMMs, but HMMs are time series models. They don't know anything about space. There's nothing spatial in an HMM, the kind of standard HMM. Yeah, they can't be used to make any inferences about space. So after that whirlwind introduction to hidden Markov models, I'm just going to run you through 4 projects that I've worked on with HMMs and movement data. So the first one is HMM for fisheries risk and marine protected area use. And this is a big collaborative project that involved white shark scientists, marine spatial planning experts, and me doing the analysis. The next project is using HMMs to understand spatial learning in hummingbirds, and that was a really interesting project, and I'll tell you more about why when I get to that. I also worked on HMMs for accelerometer data and HMMs for diving data in seals. So here we go. So we had data from white sharks, and we wanted to understand where they were going and how they were interacting with risks like fishing and protection like marine spatial planning areas. So we had locations, we had tags, I'll talk about tags in the next slide. We had locations, X and Y locations from satellite tags, and we turned them into step length and turning angle. And this is a really standard thing to do with location data. And we looked at interactions with fisheries and spatial management in the Southwest Indian Ocean. And the information we had about individual sharks was sex and size. And we also had spatial information and temporal information about where they were when from the location data. So this involved 33 white sharks that were tagged in 5 locations. There's some little numbers along the coast of South Africa, if you can see my cursor. And these are the places where the white sharks were tagged. It was a range of males, females, juvenile, subadults, and some adults as well. And the tracks, each color represents a different individual. And as you can see, so this is the black line around the coast of South Africa is the exclusive economic zone, and that's where we had fishing information. But as you can see, sharks ranged far outside that into the Southwest Indian Ocean. So the questions we wanted to answer with using this work was, what kinds of movement do sharks do? Because we didn't have that information. Does movement vary with individual characteristics, sex and size? And what are the implications for movement? The implications of movement for spatial management. So we had irregular locations, as many of you will with location data. We regularized those tracks using a correlated random walk model in, it used to be called foie gras, and now our package is called Animotum. So we had step length and turning angle as our state dependent variables. We only picked 2 states because we don't know an awful lot about white shark movement, so we wanted to keep it simple. And the covariates we had were length of the shark, sex, and the day of the year. And we fitted the model using the momentum package in R. So this is what the state-dependent distributions looked like. We had the resident movement state, which was slow and undirected. So when this turning angle is very peaked, it means it's very directed movement. So this resident state had a less directed, had a less peaked turning angle and was less directed. So when we plot the states on the map to see where these different states are occurring, we see that the resident movement is happening really close to the coast in both males and females, and the transient movement is happening all across the Southwest Indian Ocean away from the coast. But it's also quite a lot of it's also happening at the coast. So it isn't a strict separation. If we look at how life stage across the day of the year, you'll see that in the top left, the adult females and the subadult females are the only 2 life stages where the animals are more likely to be transient throughout the year. So the green line is transient. So they're much more likely to be in transient behavior throughout the year. If we look at the adult males, they're only more likely to be transient in the summer. In the winter, these 2 confidence intervals overlap for the resident and transient movement. Then the subadult males and the juvenile females have a very similar picture. They're more likely to be resident in the winter months, but equally likely to be resident or transient in the summer months. Finally, the juvenile males are the only ones where they're always more likely to be resident than they are transient. And this gave us really interesting and really important information about the risks that these animals are facing. So what we learned is that we have this coastal residence offshore transients, separation of males, and the smaller animals are more at risk from fisheries because those are happening closer inshore. We also found that the gear is the main driver of catches, and I haven't spoken about that here, but you'll have to have a quick look at the paper to see more detail about that. And we also found that marine protected areas see higher than expected use. So here's an example of using a fairly standard HMM to gain insights into how these animals are affected by risks and also by protection. So next up is HMMs for spatial learning. Sorry, I was checking my watch there. So I was involved in a project where we were able to actually track individuals without having tags on them. And this enabled us to look at how animals build up spatial memory. Because we've got these 2 different fields. Comparative cognition generally works with experimental lab studies. So that's how you tend to test things in the field of comparative cognition. Whereas in movement ecology, we tend to go out and do observational studies. But Because we were able to work on this study system, which involved hummingbirds, we were able to combine the experimental aspects of comparative cognition with the field elements of movement ecology and modeling to find out how spatial memory builds up in these hummingbirds. So hummingbirds were a really good model system because they feed often, they have excellent spatial memory, and they're readily trained. They're quite territorial. So all of this combined meant that you were able to train hummingbirds to come to a feeder and feed, and you knew they weren't going to leave the area because they're territorial. So this is what the study site looked like. They trained 14 male rufous hummingbirds with landmarks. The landmarks are these two red cubes here in the bottom left of the picture. And then they trained them with the landmarks, and then they removed the landmarks with some groups, and they recorded 3D flight paths using video, these two video recorders here in the picture. So no devices were attached to hummingbirds, and that's really neat because hummingbirds are very small, and it also didn't disturb their behavior. It also meant that we don't actually have any error or very negligible error. So in the first experiment, the birds were just naive. They flew in, they found the flower, it was just one trial involved. In the second experiment, they moved the flower to a different location, and they removed the landmarks for some birds. And in the third experiment, they allowed the birds to repeatedly feed from the feeder and learn its location. And some of the birds had landmarks and some didn't. So the benefits of an experimental setup is that we know what the motivation is. They learn to feed from a feeder, and then in the experiment, they removed the feeder, so the birds were looking for it. The benefits of video capture is that we don't have location error, and we can also choose the time interval between locations. Here we chose 1 second because the birds move so fast. So we had 3 movement variables. We had step length, pitch, and yaw. Pitch is nose up and down, and yaw is nose side to side. So our questions were, what kind of movement do hummingbirds do? Does movement vary with distance from the experimental flower that they learn to visit? And how does landmark presence and experience affect searching behavior? Again, we fitted this using momentum. So again, we had two-state models, and this is typical when you don't know a lot about the system. It's more parsimonious to just have 2 states and then increase from there if you see that 2 states aren't capturing the movement adequately. So the search behavior had a very small step length, so it was slow, and it was also very convoluted. So we've got these peaked. We have flat pitch and yaw, so it's undirected, whereas a travel is faster and more directed. So what did we see in 3 experiments? In the first experiment, we had 1 training trial, and the landmarks were present for all birds. And we found that what you're seeing here is the probability of search on the y-axis going from 0 to 1. And on the x-axis, you're seeing the distance from flower. So the closer you are to the flower, the more likely you are to be in search behavior. And that was really clear for all of the birds. They all had the landmarks present. In the second experiment, where they removed the landmarks for some birds, they found that for the birds that had the landmarks in the red line, the probability of search was highest, closest to the flower. But when the landmarks were removed, they were equally likely to be searching at any distance from the flower. In the third experiment, where animals were actually had experience of where the flower was, it didn't actually matter whether there were landmarks or not, because the animals had learned where they were. So in summary, in the first experiment, where the birds were all naive, searching behavior was focused on the feeder. In the second experiment, where they removed the landmarks for some birds, searching behavior was focused on the feeder, but only when the landmarks were present. And in the third experiment, we found that searching behavior was focused on the feeder irrespective of the landmarks. So the landmarks weren't really being used anymore, other cues were presumably being used. So what we found was in the experienced birds is that the probability of searching didn't change, but what searching actually looked like in terms of its distribution did change. So travel became slower with the landmarks present. So what did we learn from this? We learned that birds use visual cues to find the profitable locations involved with the feeder. We have evidence of learning even after one single visit, but also that other cues are learned after multiple visits to a location. And we also found that if we combine experimental design, field study, and modeling, we can actually get at a functional interpretation of movement, because unlike in field studies, we actually knew what the bird was looking for. This is, if you want to read more about it, is on bioRxiv as a preprint. Okay, so this is number 4, application number 4, and this was HMMs for accelerometer data. So I was involved in a project looking at eagle behavior. We had whole body acceleration from tags that were attached via a harness. And whole body acceleration is interesting, and it measures activity in a meaningful quantitative way. So it tells you what kind of activities the bird's doing, whether it's standing, whether it's flying. Common aims when using accelerometers on animals is to identify known behaviors or movement modes and relationships with covariates, and that's what we were also trying to do. So these tags were attached to Verreaux eagles in South Africa, and we were looking to link movement characteristics to the conditions experienced to describe patterns in movement and extract information about covariates. The raptors aren't cryptic in that they're quite large generally, but they're still hard to observe because they range really widely. And they also use upward lift of wind currents, and that depends on both topography and weather. So those are the covariates of interest. So Verreaux eagles are large cliff nesting raptors that occur in sub-Saharan Africa. Verreaux eagles rely heavily on low-cost subsidized flight because they're very big, and they do 2 types of soaring, mainly thermal and orographic or ridge soaring. So they'll soar up and then tend to glide down and do some feeding and some hunting, and then they'll find a thermal or a ridge line and do the same thing. They're really easily observable near the nest during nesting, but they range really widely. And data was collected using solar-powered GPS tags that are downloaded to a ground station that was set up in front of the nest. So whenever the bird returned to the nest, it would download all its data. So this is as close as I got to an eagle. This is a little black speck in the middle. I went on field work, but we didn't get very close to them. So these are the data that we had. These are the GPS locations, and the accelerometer data I'm going to show you come from the bird that's in a black, number 722. This is the west coast of South Africa. Cape Town is not too far from here. It's about a 3-hour drive. So this is what it looked like. We didn't have information at night because the birds roost and the tags are programmed that way to conserve battery. So I'm just going to show you analysis of this one day. And you can see that below here, so that there are these big spikes, and on the y-axis, this is we collapsed. So we had data in the 3 axes, and we had 20 points per second, and we collapsed those down into minimum specific acceleration, which separates out the static component of movement, just the gravitational component, from the movement itself. So we didn't want to remove the gravitational aspect and just keep the movement, the dynamic aspect. So our questions were, what are the parameters of the component distributions? What are the transition probabilities between states, and how are these related to topography? So we built a model for activity based on acceleration, and we implemented it in R from scratch in a maximum likelihood framework. So you end up with a lot of data with accelerometer data. So we had 84 days of data from one bird, and this was about 40,000 observations. So again, there's a bit of a trend here. The next example, if it's something other than a 2-state model, but it tends to be better to start simple. And when you don't have a lot of information about what's going on, or you can't detect patterns in the data, 2 states are a good way to go. And the covariates that we put in the model were terrain ruggedness index, and this tells you something about how what the landscape is like below where the bird is flying. So when the ruggedness index is low, it means it's over a flat area. When terrain ruggedness index is high, it means it's over a mountainous rugged area. So we fitted the model to the minimum specific acceleration data, and we found that we had these 2 states. The one had very low minimum specific acceleration, and the other one had slightly higher. And with respect to terrain ruggedness index, we found that the probability of finding the bird in this higher acceleration state decreased with terrain ruggedness. So you were more likely to have a bird in a high acceleration state over flat areas. So sorry, this in the first line where it says the Viterbi decoded states, this is just showing that the bird spent 45% of its time in state 1 and 55% in state 2, so about the same amount of time in both states. And just what I explained from the figure, we found that low acceleration flight is more likely in flat areas, and flat areas tend to generate thermals. So that was interesting to know, because it seems like thermals could actually be really dynamic structures, but perhaps the thermals that were generated during the time that the bird was collecting data was in a season when it wasn't windy and you just had these sort of thermals rising up without a lot of turbulence in them. So that's interesting as well. And this was published back in 2017 in Methods in Ecology and Evolution. So last but not least, this is an example of work that I did using diving behavior. So far I've talked about animals just moving on the ground or just in 2D in the sea. But here I actually had information on the diving behavior of these seals. So this is a picture of a Weddell seal with a Sea Mammal Research Unit instrumentation tag on its head. These tags last about a year until, or normally less than a year. We put them on right after the animal's molted, so it's got a fresh coat of fur, and it'll last until some months, normally before the next annual molt. So that's a year is the maximum amount of time that it'll stay on. We tagged these seals in the Weddell Sea. So this is the Antarctic Peninsula. You can see it's pointing out into the Southern Ocean, and the Weddell Sea is a big embayment just to the east of the Antarctic Peninsula. We tagged 19 seals in this area in 2011. Weddell seals are large-bodied and they're long-lived, and they live in the pack ice all year round. They dive really deeply, and they also hold underwater territories. So there was interest in finding out how they're using these different depth layers in the water column. So this is what their environment looks like. Lots of ice flows as far as the eye can see, rich and sort of diverse underwater sort of topography in terms of all the ice flows. This is what their diving behavior looks like. So we've got depth on the y-axis. So these dives are going down to about 350 meters, and time of day. And this is just a few hours in the dive record of one animal. So the Weddell Sea physical environment is quite unique for Antarctica. It's got this really wide continental shelf, which is unusual, and it's got really extensive winter sea ice. And it's also special oceanographically because Antarctic bottom water forms here. Antarctic bottom water is a very cold, dense water mass. It forms here and it actually sinks to the bottom, and it fuels global ocean circulation. So it actually contributes to the Gulf Stream that we experience in the, you don't experience on the West Coast, but on the East Coast of the US and the West Coast of Europe, we experience it. So this is the sort of yellow, lighter color is the ice, and then the really dark blue color is very deep water, and then you've got the continental shelf instead of green. And all of the data we collected are happening in this right-hand corner of the continental shelf, and you can see there's a bit of a trough here, and the seals really like that area. So we've got this seasonally varying, really highly stratified by density water column. So imagine sort of oil and water. You've got these really defined layers in terms of density in the water. So we've got these, we call them water masses. If you're familiar with these oceanographic terms, there are the variety of available water masses, and they really are sort of little parcels of water, and there are quite hard interfaces, not hard, but there are defined interfaces between the water masses. So these are the tracking data. The males are in this darker sort of purple color, and the females are in pink that go out into the really deep water. So you can see there's this trough underneath, which is a bit of a deeper area. So the data that I analyzed were between February and June 2011, and we had 10 females and 9 males. Again, location, we regularized the location data using this package by Ian Johnson. And we wanted to know what kinds of dives that the Weddell seals actually carry out. Can we group them? Are certain kinds of dives linked to water masses? And is there a seasonal or diurnal pattern to the diving behavior? So I fitted 2 HMMs, one to males and one to females, female data, because it turned out, I tried to fit them all together, but it turned out the data were really different. So I fitted the same model, but almost the same model, but to males and females separately. So the state variable, so in a normal vanilla flavored movement model, you tend to have step length and turning angle as your state dependent variables. Here, I wasn't interested in modeling the horizontal behavior, but rather the vertical behavior. So I picked aspects of dives and used those as the state dependent variables. So here we had duration of the behavior, so duration of the dive, the hunting depth, and we defined the hunting depth according to some dive parameters. The proportion of time, the portion of the dive spent hunting, the proportion of the bathymetry reached. So if they reached the bottom, that was 1. If they reached halfway to the bottom, that was 50%, that kind of thing. And also we looked at the salinity at hunting depth. And we could do this because the tags also collected temperature and salinity of the water that the seals were diving through. So we had all these aspects of the dive and we wanted to estimate the underlying states. So because these seals spend a lot of time either doing quite shallow dives, you can kind of see them in the dive record. You can see these really shallow little blips. And they also spend time hauled out, just lying on the ice resting. So we knew those behaviors happened, but we weren't interested in estimating different types of haul out or surface. So we actually put them into the model as known states. So the model didn't have to estimate those. But we did want to estimate the different kinds of dive types, so we estimated them. And the covariates we had were a local time of day and week of the year. A week of the year is important because in the Antarctic, or at least the latitude that these guys occur, in the middle of winter, there's no light at all. So the sun dips below the horizon and doesn't come out again for a few months. So a week of the year was sort of a proxy for light availability. So this is an infographic that I developed together with a scientific illustrator to illustrate the findings of this work. So we found that males and females share a number of dive types. So each of these little seals has a male or female symbol on it. So here we've got males and females falling out in the ice and diving shallowly under the ice. We also have males and females diving on the continental shelf, deep down and feeding benthically. But we only have the females going out into this pink water, which is actually what's meant to show that it's warmer water, and feeding pelagically. We found that both do deep dives into this cold bottom water here on the benthic areas, but only females do these pelagic dives into warmer water masses. So this is, if you're at all familiar with oceanographic temperature salinity diagrams, this is what this is. So basically you take the salinity on the x-axis and temperature on the y-axis. So this is temperature and salinity of the water where the seals are diving. And it's actually at the bottom of their dives, the deepest point of their dives. And you can see that, so these are colored by the state that the HMM estimated. So the shallow, we have these shallow and epipelagic just in the first maybe 10 to 50 meters of the water column. These shallow dives, and they're occurring in this part of the diagram where we have fresh water. Basically, the salinity is low, we've got fresh water because there's lots of melt input from ice. Then you have these deeper dives. The benthic dives, unsurprisingly, are happening in these really cold, dense water masses that occur on the bottom in the benthic areas of the Weddell Sea. And on the left, we've got the female map and on the female diagram, and on the right, we've got the male diagram. What you'll notice here is that in the female diagram, there are these dives, these points in beige, and these are basically dives that are happening into water that is a mix of two different water masses, and it's got a very characteristic signature. And this acronym here it's called modified warm deep water, and it is warm compared to the surrounding water, and it's got input from offshore water masses. So it's a mix of the really cold water that's forming on the continental shelf and these warmer water masses that are offshore. And it shows that females are going into a very different environment to the males. None of the males really did this. We can't really see any of this sort of line shooting up here. You can see the beginning of it here, but none of the males did dives into really into water that was much warmer than 0 degrees. So only females do these pelagic dives into the warmer water masses. And we also found that in this, you can see in the little map in the left-hand corner, bottom corner, you can see that the male locations are in yellow. So the males stay on the shelf almost exclusively. They don't really venture off the shelf into the diving over deep ocean. Whereas the females also leave the shelf and dive pelagically. They also do deep dives as well, but they also leave the shelf, which the males don't do. This was published a couple of years ago in Proceedings of the Royal Society to find out more. So I hope with this brief intro and these 4 examples, I hope that I've convinced you that HMMs are really versatile. You can use them for lots of different things. They can't do everything, but they are really fun and you can use them in lots of different ways. I think that's it. And thanks very much.
Brilliant. Thank you very much, Theoni. So just checking, is there any questions from anyone in the room? Or if anyone online wants to put any questions in the chat? Yep, perfect. Yeah, we've one question in the room here, Theoni, if that's good with you.
Sure.
So thank you very much for your presentation. It was very clear. Do you have like the equations of your latent space? Because when estimating with TMB, this constrains the latent space to be Gaussian, and this can be quite constraining. And I was wondering how much it was constraining for HMM. Is my question clear?
Yes, I think so. It's relating to TMB, right?
Yeah, and to the formulation of your hidden space.
So I haven't actually used HMM TMB, that package yet. I've only used the momentum packages and written the HMMs from scratch. Momentum's really useful for just quickly fitting models, and if you want to fit lots of models. So I wondered if you could please repeat the bit that was not relating to TMB.
Would you have the equations? Yeah. Would you have the equations of your models that you are using, not only diagrams but equations?
I don't have them handy, so yeah, sorry about that. But the equations, so they're really straightforward models to, once you know how they work, it's a little bit like Lego. You just add, you work out the probability of each of the distributions for your observation, and then you add them all together. So probably the best resource for going more deeply into how these models work is the Zucchini, McDonald, and Langrock book. It was published in 2016. It's a hidden Markov model book. So I don't have the equations of my models handy. All of my papers have code associated and appendices that have methods in them, but I didn't include them in this talk. I thought it was probably not quite the right audience for just a quick talk with equations in it.
Yeah, thank
Brilliant. So Theoni, I just had one quick question about, I haven't done any work with HMMs, but when you're setting them up and deciding, when you're setting up the model and you're deciding how many states to explore or to include in the model, is that being determined by the questions that you're asking, like by the hypotheses you have, or what you know about movement behavior of the species you're looking at, or kind of a mixture between the two? Like, what's the kind of best practice for that?
So typically I would start looking at the data first and have a look. So you want to have an idea of the system and knowing, well, how many states do I think exist in these data? But mainly you'll be looking at the data and saying, well, how many states can I see? You might think like, oh, these animals do this kind of behavior, or like say they do 4 kinds of behavior, and you think you might be able to see that in the movement data. You might not. You might just be able to detect 2 states and the rest is sort of hidden in there. But in other cases, you might be able to see more than 2 states. I would always start out with 2 states because it makes it easier to find starting values, because you need to provide starting values to the optimization algorithms. Yeah, there's a lot that I didn't talk about in this talk, but the starting values are really actually an important part of fitting your HMM. You need reasonable starting values, otherwise you might not have convergence. So yeah, I would have a combination of arriving a priori with an idea of what you think is happening in your system, but also primarily looking at, once you're looking at the data, saying like, okay, I thought I could detect certain behaviors, but actually the data don't support that at all. And it's always worth being conservative with states, because you need to be able to interpret them for your results to make sense. And often it's hard to interpret. And also, if you think about it, basically, the more states you have, you're segmenting your data and picking up on more and more and more detail. So it sort of depends on what your question is, as you said. So if you're looking for overall patterns, then starting with 2 states is a really conservative, like a good way of doing it. Whereas if you're like, well, I want to detect like a really rare behavior, then you might want to add states until you can pick that up, if that makes sense.
Hello?
Hello? Sorry, I didn't. Did you hear me? Did you get that?
Yeah,
did you get that answer?
Audio gone again. No. And then, sorry, we just have a couple of questions in the chat. So from Richard Townsend, I hadn't thought of the covariate plots versus location. Do you use this often to show the relationship?
Thanks for the question. Yes, so if I have, so even though the location data aren't going into the model, you have those data. So you can say like, okay, where are these states happening in space? You're not using space to inform the model at all, but you're just visualizing the states in space. Yeah, I do that really often.
Brilliant, thank you. And then just one more from John W. Was weather considered as a covariate for the Eagle study?
So it was, but it wasn't very good. So the acceleration analysis was sort of a proof of concept. So we didn't do that for all of the birds, but there is a separate study that we did for all of the birds where we didn't actually look at the acceleration data, but we did look at the GPS data. And because we had XY and also elevation, we could see if the birds were gaining height and if they were spiraling. So we could see if they were thermaling that way. And we did have weather information, just the nearest weather stations, because these birds are in quite remote areas. The nearest weather station was really far away. So it wasn't a very good reflection of the conditions that the birds were actually experiencing. And that's quite a tricky thing to get around, because these birds are exploiting sort of really localized weather conditions and also just wind patterns and yeah, you know, the thermal formation depends on all sorts of things, and we weren't capturing those things. But we were interested to know, well, is thermaling more cost effective than ridge soaring? if you're using less acceleration, basically means less activity. So if a certain type of soaring involves less energy, then that's something that's really interesting to know. We haven't quite managed to get that yet. Thanks for the question.
I'm muted again. Brilliant. Okay, well, thanks a million, Theoni. That was a really, really fantastic talk.
Thanks for the invitation.
And yeah, and thank you. We know that time difference can be a bit of a nightmare. So yeah.
Actually worked really
Thanks
well, because I've got a small child, so he's in bed, so that worked really well for me, actually.
Perfect. Yeah. Brilliant. Great. Okay. Well,
great. Well, thanks a lot. Bye.
Bye now. Bye bye. How do I save? Ernst, do you know anything about recording Zooms? If I just stop recording, will it just store it?
And then I think it will automatically save.